everyone, and thank you for joining us today. I'm thrilled to see so many regulars in the Zoom room, as well as some newcomers. Welcome all. For those of you who don't know me, my name is Mara Jean Tilley, and I'm privileged to head up the Garvan Research Foundation, which is the marketing and fundraising arm of the Garvan Institute of Medical Research. And I'll be your MC today for Bite Size Science. As some of you will know all too well, being told you have cancer is distressing for individuals and their families. Being told that your treatment has stopped working is simply devastating, but regrettably all too common. In Australia, treatment resistance causes 1 in 3 cancer deaths. Dr Arcadi Cipponi has been studying this question for more than 7 years, and I'm excited to report that his dedication, passion, and determination has recently led to a major breakthrough, uncovering the mechanism that allows cancer cells to evolve and become resistant to our best cancer treatments. Before we kick off, I'd like to pay my respects to the Gadigal people of the Eora Nation, the traditional owners and custodians of the lands on which the Garvan Institute and St Vincent's Hospital are located. We recognise their continuing connection to land, waters and culture, and pay our respects to their elders, past, present and emerging. A little housekeeping. Due to time restrictions, we will not have a Q&A today, a live Q&A that is, but we will have the opportunity for you to vote on some questions, and the winning questions will be addressed with Akari. If you have questions or comments that are not addressed throughout today's presentation, please do enter them in to the survey at the conclusion of the seminar, along with your email address, and somebody in the team will be sure to come back to you as soon as they can with answers to your questions. Now, it's my great pleasure to introduce Dr Arcadi Cipponi, who I've had the delight of working with for a number of years here at Garvan. Dr Arcadi Cipponi is a Senior Research Officer in the Genomic Cancer Medicine Laboratory here at the Garvan Institute. His research is focused on the characterization of common genetic and molecular mechanisms that enable cancer cells to evade drug treatments. This is what we're talking about today, drug resistance. Dr Cipponi completed his PhD in molecular biology at the University of Milan, Italy. Arcadi, welcome. So,
thank you, Margin. Hello.
Hello, it's lovely to see you, albeit via the Zoom camera.
Yes.
Now, tell me, how did you become interested in cancer resistance specifically?
Okay, so, well, what I would say is that the acquisition of drug resistance always requires extensive genetic and molecular changes in a cancer cell. And so it's extremely interesting to understand the dynamics that regulate these chains. And the second reason I would say that, as you just mentioned, drug resistance is a major problem in medical oncology, and it's extremely important for us to understand the underlying mechanism, to have a deep understanding of what happens, what drives drug resistance. Because if we understand this mechanism, we can target some of these specific molecules and increase the response to anti-cancer treatments.
Thank you, Akari. And just so I understand clearly, essentially we're talking about a mechanism whereby a treatment may have worked initially and very effectively, but it's the cancer cells themselves that are evolving so as to be resistant to that treatment that originally worked.
That's correct. It's exactly like this. Cancer cells, initially in a cancer, most of the cells are very sensitive to the drug, and anti-cancer treatments kill most of the cancer cells initially. But in some patients, there is a subset of cells that I will describe shortly that are intrinsically resistant, or they are able to acquire very quickly the resistance to the drug. And this is what we have been studying for several years. What are the underlying molecular and genetic mechanisms that these cells utilize to adapt and evolve in response to drug and ultimately to restart proliferating and inducing the relapse of the cancer.
Scary stuff. Well,
Yeah.
let's get on with your presentation, Arcadi. Thank you so much.
Okay, I'll share my screen. Okay, so good afternoon, everyone. Thank you very much for tuning in. I have to say it's a great pleasure to have the possibility to share with you some of the results of the studies that we conducted here at the Garvan Institute. As a general introduction, I will say that human cancer is a very complex disease. It's characterized by an incredible genomic instability. This means that cancer cells are continuously accumulating new mutations. And this generates an enormous genetic diversity. For example, if we take different patients with the same cancer type, every single patient would have a different genetic profile, would have private mutations that make it different from the cancer of another patient. Now, because of this extensive genetic diversity, we always, as Margine mentioned, we frequently see the acquisition of drug resistance. So there is a negative impact of the genetic diversity on these anti-cancer treatments. And in this example, a breast cancer patient received surgery. The cancer remained stable for a few years, but after cancer cells started to proliferate, she was treated with chemotherapy, the first pharmacological approach. Cancer cells responded very well, but then they started to grow and they were not responding as expected to a second pharmacological treatment. Now, the traditional model that we utilized to explain this clinical response was that one of what we call passive selection. In other words, in response to anti-cancer treatments, most of the cells are killed, but a few of these cells have an intrinsic resistance to the drug, or like I said before, are able to quickly adapt to the drug very quickly. And so essentially, these cell subpopulations are selected and they are able to quickly restart growing because all the other cells have made lots of space and they are now able to thrive in the presence of the drug, unfortunately. So a couple of years ago in our lab, we hypothesized a second possibility, a second model of resistance. So cancer cells, when we treat cancer patients with targeted anti-cancer agents, and so these are drugs that target specific molecules in a cancer cell and do not directly damage DNA. They simply say to the cell, stop dividing. So the idea was that in response to this specific class of therapies, cancer cells could increase their genomic instability. In other words, they could start to mutate even faster. And this is why we call this model accelerated mutagenesis. And so it was very interesting because we knew that similar mechanisms are present in microorganisms, for example, in bacteria and in yeasts. And when microorganisms are exposed to environmental or pharmacological stress, like, for example, antibiotics, or they are in the presence of lack of nutrients, of lack of oxygen, these cells become hypermutable. They increase their mutation rates. And this strategy has been evolved because most of the cells would acquire lots of deleterious mutations, they would die. But a few of them would acquire the beneficial mutations that would help them to survive and adapt in the presence of the stress or the drug. And so we said, would it be possible that similar mechanisms would be present in human cancer cells? And the presence of these mechanisms is extremely interesting and important because if these mechanisms are present, we can target this mechanism to stem the generation of the genetic diversity and therefore the progression of a cancer. Now, in our lab, we immediately started to set up different experiments using in vitro models of resistance. And in these models, what we've done is to take different cancer types and recapitulate essentially what we see in a patient. we killed most of the cancer cells with a drug, and we saw that over weeks and months, almost up to a year, we saw that a few cell subsets were able to survive the drug treatment and progressively increase their fitness in the presence of the drug. And what we've done is do extensive profiling of these cells over time. We studied the molecular mechanisms and their genomic profiles. How did they evolve over time? And I would like to share with you one of the most striking results we had at the very beginning of the study. What you see here is a microphotograph obtained using a confocal microscope. It's a microscope essentially which is able to identify fluorescence. The red dots, the red circles that you see, the red area are the nuclei of cancer cells, and the green dots are clusters of proteins. These proteins are recruited to repair DNA damage. Now, like I said, these drugs don't induce DNA damage. They don't directly mutate the DNA as chemotherapies, for example. And so we were very surprised to see these levels of DNA damage in these cells. If this damage is not repaired efficiently, it can induce lots of mutations. So this is exactly what we have done, to look at the acquisition of new mutations in these cells when they were exposed to these targeted therapies. So strikingly, what we saw is that the resistance cells are the letters and numbers that you see in these graphs, were acquiring lots of mutations compared to the untreated cells. What you see here are what we call them phylogenetic trees, but it's a way to, say, describe the genetic diversity, the genetic distance between two populations. As you can see, the black letters and numbers are the untreated cells, and the red ones are the treated cells. As you can see, they cluster very well, and they are very far apart. This means that the drug-resistant cells were accumulating lots of mutations that were not present in the untreated cells. And so the next experiment that we set up was essentially trying to answer the question, what are the underlying molecular mechanisms? We need to know what is driving this dynamic change of mutability in cancer cells. And so what we've done is to take a cancer cell line exposed to different pharmacologic pressure, different target anti-cancer drugs, and then we silence every single gene in the genome. There are almost, well, let's say more than 20,000 genes in our genome. So this was a massive experiment. And so what we've done is then to identify the genes that were responsible for this dynamic change of the mutation rates. And the key genes were a group of proteins, let's say, that are known as Entor signaling. So Entor is a group of proteins that essentially is able to sense the stress in the cell. And the stress could come from environmental conditions like, again, lack of oxygen, the presence of reactive oxygen species, or lack of nutrients, or inside the cell from DNA damage, for example. And a very important piece of this puzzle was to understand that Entor signaling was communicating with other signaling pathways, other specifically DNA repair pathways. And what was very interesting to understand that when Entor was sensing the stress coming from the DNA damage, it was telling to the DNA repair pathways to stop working or stop working efficiently. And so cancer cells were deliberately acquiring new mutations. They were replicating their DNA in a, let's say, in an unfaithful way, in a sloppy way, if you want. So by doing this in the presence of stress, they were acquiring new mutations. Now, one of the first experiments that in science one of the first steps that in science we always take is that to validate the results, try to repeat these results in the same model, but also across different models of resistance. And this is exactly what we have done. So what we've done is to silence Entor in a specific cancer, in this case it was a melanoma, and expose these cells to a target drug or left them untreated. So in this graph, what you see on the x-axis on the bottom is the cell proliferation. On the left side, you see the white bars. Now, cancer cells, when we silence Entor, they reduce the proliferation, as expected. We know this very well. But what was surprising is that when we were silencing Entor in the presence of the drug, so the right side of the graph, the gray bars, cells were behaving exactly the opposite. So the silence of Entor was fostering the proliferation of the cells in the presence of the drug. Obviously, like I said, we cross-validated these results in other cancer types, in breast cancer, in liposarcoma, which is a cancer of soft tissues. Now, there are a few important things that I would like to say. Essentially, what I just showed to you happens when cancer cells are treated with target anti-cancer agents, and they're able to switch to this hypermutable state to increase the chance to pick up beneficial mutations that foster the adaptability. Now that we know these mechanisms, we can think about different strategies to target these mechanisms, and so to stem the generation of these new mutations and make sure that cancer cells are responding better for a longer time to the anti-cancer treatments. So I mentioned DNA repair. To give an example about one of the strategies that we are thinking, in this slide, I'll show you a cancer cell which is using two different DNA repair mechanisms, A and B. Now, when we expose cancer cell to a target agent, anti-cancer agent, like I said, what happens is that one of these DNA repair pathways is repressed. It becomes less efficient. Now, in this situation, cancer cells become heavily dependent on a second DNA repair pathway, the pathway B. So if we target that specific pathway with an inhibitor, we induce a lethal mutagenesis, as we technically say. In other words, cancer cells acquire so many mutations that it cannot tolerate these levels of mutagenesis. So essentially, the idea is to now treat cancer patients with a combination of drugs. One, the anti-cancer treatments, which targets the oncogenes that drive the proliferation of the cancer. And then add a second drug, which targets a specific DNA repair pathway. Now, a quick summary. We have seen that cancer cells are genetically unstable. Genomic instability drives the resistance to anti-cancer treatments. We have seen that, like in microorganisms, human cancer cells can accelerate the mutation rates in response to specific pharmacologic pressures. We have seen that Entor signaling, a group of proteins that senses stress in the cell, is able to facilitate the acquisition of drug resistance by repressing the fidelity of DNA replication and repair. It's also, we have seen how we are thinking now to utilize these strategies to increase the activity, the effect of anti-cancer therapies. As Marajin probably mentioned at the beginning, this has been a massive project here at the Garvan Institute, and we had a lot of collaborations with other institutes here in Australia, like the Peter MacCallum Cancer Centre and the Walter and Eliza Hall in Melbourne, and also other research centers overseas. I have to thank David Thomas, Professor David Thomas, who has been leading these studies, his incredible support, his inputs. He has been fantastic over these many years. And I also would like to thank the Gergenson Foundation for donating funds for these studies. I really have to thank Ingeborg and Thomas Gergenson for their generosity and their support during these years. Thank you very much for listening, and happy to take your questions.
Thank you, Akari. You should feel very proud. It's an amazing piece of work and recently published in a very prestigious journal. Akari, you mentioned DNA pathway drugs. Do such drugs already exist, or is this something that will need to be developed?
That's a very good question. Many of these drugs are already in utilized in cancer patients. And these drugs are used in a very small subset of cancer patients. And the good thing about this discovery is that we will be able to extend the use of these combination strategies in different types of cancer and different patients. I don't want to go too much into the details, but yes, these drugs are ready to be used, and we are really now thinking to design new clinical trials to test these combination strategies.
It's really exciting, Arcadi. Now, for our audience members, you will see that the questions have been brought up for you to vote on. So looking at poll number 1, you've got a choice between what are the next steps for Arcadi's research, or how will advances in genome technology impact on Arcadi's work. Going to poll number 2, you can vote for, is it surprising to see stress-induced mutagenesis, forgive me, in cancer cells? Or is it possible to detect if a patient's cancer could develop drug resistance before it actually happens? Poll number 3, you can vote between how long do you think until Arcadi's research could be an option for people facing treatment resistance in cancer? Or are there any cancers your work will have a better impact on than others? So just give the audience a few more moments to respond to those questions. Arcadi, you have already spoken about the length of this particular project, and more than 7 years in the laboratory. And from what I understand from Professor David Thomas, a lot of the experimental work was completely novel, and you have personally crafted many of the technical capabilities to be able to do this research. What gets you out of bed every day for 7 years to achieve this incredible breakthrough?
Yes, well, I would say that obviously, first of all, the passion to understand this molecular mechanism, but also the challenge, both intellectual challenge and technical challenge. Our job is very different, I would say. Every day we face problems, technical problems, problems really where you don't understand what the results that you have. It's difficult sometimes to have a good interpretation of the results. So you need additional experiments to understand what you got 2-3 weeks before. And so it's a continuous challenge for us, but this makes everything also very interesting. And I would say this is what the driver for me, but certainly for all my colleagues here at the Garvan.
Yeah, thanks, Arcadi. And you mentioned the Gergenson Foundation.
Yes.
I think we might even have Thomas and Ingeborg in the audience today. Hello, guys. What's the impact of their philanthropy been on your work and on the speed at which you've been able to conclude this epic experimental model?
Well, I certainly have to say that thanks to their support, we have been able to perform additional experiments. Science is extremely expensive. Thanks to their support, we have been able to purchase reagents for the whole genome RNA interference screen, which is essentially one of the slides that I show. That experiment lasted for more than 2 years. And it took us 2 years to come out with this Entor signaling. And their support has been key to keep going with this experiment. And I also have to say, during these years, it's been such a pleasure to catch up with Ingeborg and Thomas and update them on the latest results. It's been lovely, and I really enjoyed my meetings with them. And yes.
Yeah, it's really special, isn't it? We've got many donors and partners for the future in the Zoom room today, and we hope you know just how impactful your support is, and it goes beyond the philanthropic support, as Arcadi says. Having the enthusiasm and the commitment and the insights that our Garvan family members make is such a privilege. Alrighty, we've got the results on the poll. So question number 1, Akari, what are the next steps for this piece of work?
Well, so the next steps are certainly to test these combination strategies in different types of cancer. Although our results really are strongly suggesting that these mechanisms are common across different cancer types, we tested in breast cancer, melanoma, liposarcoma, and pancreatic cancer. We need to understand whether other cancer types are also, we can target other cancer types with the same combination strategies. But certainly, another line of research is to understand whether there are molecular signatures that are able to predict the sensitivity of a cancer to these combination strategies. Again, I don't want to go too much into the details, but we can identify the expression of some genes that are indicative of that may point to the sensitivity of a cancer to these specific drugs that target DNA repair pathways.
Arcadi, I think that's a fantastic segue to the next question, which is whether it would be possible to detect if a patient's cancer could develop drug resistance before it happens. So presumably
Yes.
before that first frontline treatment is given.
Yes, that's right. Well, the idea here, like I said, is to understand what's going on in the cancer during the first line pharmacologic treatment. When we treat a patient, when medical oncologists treat a patient with a targeted agent, then it's possible to understand how cancer cells are responding. And we can do this by profiling the cancer, or with the biopsy, or in the future, it will be possible by a simple sample of peripheral blood. And so by looking at these molecular signatures, we will be able to understand how the cancer responding to the drug, and whether it's necessary to add a second drug or simply or choose other strategies. But certainly these molecular signatures are the future in medical oncology. They will tell us a lot about how cancer cells are responding to the treatment.
And so how long before we can see this in the clinic, Arcadi? No pressure. What would the steps be to getting it into the clinic?
I think everyone is waiting for this. Genomic technologies are advancing really quickly. The near future is certainly that one to utilize genomic technology and using this peripheral blood samples, so non-invasive approach to not only understand how a cancer is responding to a drug, but also, and really this is really beautiful, to have an early detection of cancer. This is the future. And so it's better to have an early detection rather than go through a treatment of a late stage cancer where the chance of curing the cancer are very, very low. And so the most exciting thing for all of us at the moment is these approaches to identify molecular markers in the peripheral blood and hopefully go through a very early detection of cancer. This will really save millions of lives.
And so I'm going to push you a little bit on timing, which I know is a cruel thing to do. When do you think would be the earliest we'd be able to see this going into a clinical trial?
Are you talking about the genomic technology of early detection, or the combination strategies that I was talking about?
Answer both.
Okay. Oof. So for the combination strategy, it's a bit easier because, like I said, the drugs that we are thinking to use are already used in different clinical settings, but are still there. So it's relatively straightforward design a clinical trial and combine these target anti-cancer agents plus the drugs that target DNA repair. But for the second part of your question, I would say that the genomic technologies that we will need for an efficient early detection of cancer will take a while. At the moment, we have something which is very promising, but to have something that really works and can be used in the whole population, that will probably take at least 5, 10 years to really have something robust that is really reliable. But it's extremely exciting, and we already see lots of breakthrough in this direction. It's very exciting.
Thank you, Arcadi. It certainly is. Now, we're almost out of time, so if I can just shout out to our audience members, and thank you so much for attending Bite Size Science. I hope you've enjoyed hearing from Arcadi and learning a bit about his incredible work. We will have a short survey popping up on your screen shortly. Please do enter your full and frank feedback. We're still learning how to share our science in this new COVID-19 normal world, and would really value any thoughts you have on how we can do a better job. Sincere thanks to Arcadi for sharing his incredible work. A big shout out to all of the donors and Partners for the Future, board directors and volunteers in the audience. We are indebted to you and we are privileged to have your support. If you feel inspired by today's session, please do consider making a donation to Garvan's research. Please feel free to share the link to this seminar with your friends and family. It can also be accessed via Facebook shortly. The more advocates we have in the community for medical research and for the Garvan Institute, the better. Science has never been so important. Please continue to keep safe and keep well, and we look forward to seeing you at the next bite-sized seminar on the 12th of November, which will be featuring one of our talented immunologists. Arcadi, thank you once again. See you soon.
Thank you very much, Margin. Bye-bye.
Bye.